This is a brief introduction of TCPA, the Cancer Proteome Atlas portal. As you can see in the home page, TCPA portal currently contains two separate web applications for exploring and analyzing the proteomic data. One application focuses on patient cohort data, and the other is for cell line data. Let's first look at the patient cohort application. Patient cohort application includes four modules. The first one is the summary module. In the summary module, users can get the basic information of each dataset, like the number of samples and proteins. The second one is my protein module. It shows the detailed information of each protein marker, for example, the corresponding gene names and antibody validation status. By clicking the View button, users can see the protein expression levels across different cancer types in box plots. The visualization module has two submodules, network and heat map. In the network visualization, after selecting a dataset, you will get a network view in which protein markers are nodes and the interactions are colored edges. By searching a protein marker in the input box, users can also get a subnetwork focusing on the neighbors of the protein marker. In the heat map module, you can visually check the global protein expression pattern for each cancer type. In each heat map, users can zoom in, zoom out, or check the expression level for each cell and other detailed information. In addition, users can also obtain the high-resolution figures by downloading the PDF files. Now let's look at the analysis module. It has three commonly used analyses, including correlation, differential expression, and survival analysis. For correlation analysis, users can examine whether the expression levels of two proteins correlate with each other. The first four columns show the protein pairs and their gene symbols, followed by the correlation coefficient and corresponding p-value. By clicking the Plot button, you can visualize the data in a scatter plot. In the differential expression module, users can identify which proteins are most differentially expressed between any two tumor types or subtypes, and visualize the differential expression pattern in box plots by clicking the plus sign in each row of the table. The third module is survival analysis. Users can use the module to identify potential prognostic protein markers in their interested cancer types and visualize the survival data in Kaplan-Meier curves. All the protein expression data curated in TCPA are publicly available. Through the download module, users can select the datasets of interest in a tree-like menu. Now let's switch to the other application of TCPA, which is for cell line data. The user interface of this application is similar to the previous one. It has four modules listed on the homepage: datasets, MyProtein, visualization, and analysis. The datasets module lists all the cell line data curated in the database. Users can get the basic information about each cell line lineage. The second module is MyProtein. Similar to the patient cohort application, users can get detailed information about each protein in this module and can also visualize the expression pattern of each protein across all different cell line lineages. The settings of the visualization module are the same as the patient cohort application, but based on the protein expression data in cell lines. Let's skip this module and go directly to the last one, analysis. The first analysis submodule is called protein-protein correlation. In this module, users can get the correlations of different protein pairs and visualize the data in a scatter plot. The second one is protein-drug correlation analysis. Users can use this module to identify potential predictive protein markers. In this module, you need to first select a drug dataset, a cell line lineage, and the way of curing. For example, when we select search by protein, each row shows the number of drugs positively or negatively correlated with the protein listed in the first column. By clicking the Plot button, you can visualize the detailed information in a volcano plot. Similarly, you can also query by drug to find which proteins show correlations with the sensitivity to a specific drug. The last one is protein mutation correlation analysis. In this module, given a mutated gene of interest, users can identify proteins differentially expressed between the wild-type and mutant cell lines, and visualize the differential expression pattern in box plots. To download the protein expression data of cell lines, you can go to the download page by clicking the link at the top of the page. TCPA also provides a link to an online tutorial on the landing page of tcpaportal.org. On the tutorial page, you can learn how to use the two applications through the dynamic slides, which includes the video tutorials.